Welcome to your lesson on glycolysis. Now, glycolysis, you're looking at the name itself. Glyco, similar to what the word is for glucose, means sweet, talking about a sugar. Anytime you see glyco, it's a version of sugar. And then you know lysis already means to burst. Now, this is important detail here. It occurs in the cytoplasm. You do not require a mitochondria to run glycolysis. And mitochondria are the ones that require oxygen, so this is also an anaerobic process. No oxygen required. There's about 10 steps I'm going to show you on the next page. And so because it can be done in the cytoplasm without a mitochondria and without oxygen, all types of organisms are able to do this. All of them. You can take a glucose, you can split it. Again, there's 10 steps all the way down to the pyruvate, but you can take the 6-carbon glucose and efficiently split it into 2 3-carbon pyruvates and then go from there, right? I'm actually going to skip to the next page just so you can see what's going on. I'm going to go step by step through it. So here's our original glucose molecule. You can see it's got the 5 carbons in the ring and the 1 popped out. So it's a 6-carbon molecule. We've left off all the OHs, all the hydroxyl groups, and all the regular H's because it'll just bug up the actual picture. So this is just to show you the fundamental carbons anyway. So the first thing that I'm actually going to do is I'm going to want to destabilize this ring. In order to make it fall apart into 2 3-carbon molecules, I have to destabilize the bonds. And right now they're very happy to sit exactly how they are. So the first thing that we're going to do is something called primary activation. We're going to add a phosphate, and I've written it over here for you. The extra phosphate will destabilize the glucose molecule. So you add a phosphate on, and it just so happens that here's the phosphate here. It gets added to carbon number 6. So the new name for our molecule is glucose 6-phosphate. Glucose 6-phosphate, telling us the location of the phosphate on the glucose. Then you're going to do a little bit of a shift here. Now, glucose and fructose, I hope you remember, they're both C6H12O6. They are isomers of each other. But if you look here at the picture, you can see this has a 6-sided ring, and this one only has a 5-sided ring. That's because this is carbon number 1 right here on the glucose. Carbon number 1 on the fructose has been popped out of the ring. Okay? So that adding of that phosphate on there has actually caused this isomer shift. This is enough of a high energy bond destabilizing everybody else that this carbon right here is going to pop out, and suddenly you're going to have fructose 6-phosphate. You destabilize it again by using another phosphate off of an already existing ATP. It's called secondary activation. And notice that this is the original phosphate that we put up over here. And now that this carbon that's been popped out, we've added a phosphate there. This is enough of a struggle within the molecule that it's going to, in the tugging and the pulling of these 2 phosphates, that it's going to pull this sucker right apart. And it falls apart into 2 different versions. You can see here's a 3-carbon version. This one is the G3P or P-gal that we saw in photosynthesis. And then we have DHAP. We also saw this briefly in photosynthesis, but we didn't focus on it. Now, DHAP and G3P are just isomers of one another. If you were to count up all their little parts, all the C's and all the O's and all the H's, you'll see that they are identical. The difference is where you can see the difference of where we've actually got this little carbonyl group right here. So this guy is not ready to go yet, but this one is ready to be acted on by the enzyme that's going to do this job here. So this one is ready to go. The G3P is going to begin its journey down. The DHAP needs to be converted to G3P, and then it's going to follow and do the exact same process. Okay, so so far we have added 2 ATPs, stolen their phosphates to destabilize the molecule. We went from glucose to glucose 6-phosphate to fructose 6-phosphate, and then by adding that other phosphate on over here, we've gone fructose 1,6-bisphosphate. There's a phosphate on carbon 1, carbon 2, and this tells us, the bis tells us there's 2 of those suckers. All right? Please memorize those little suckers. Memorize the idea that ATP is being used here, that you're going to require 2 ATP in before you can even think about ripping glucose apart. Okay? And that your middle molecule is your G3P. It's really important that it's a 3-carbon middle molecule. So now what we're going to do is we're going to steal some electrons. Again, NADH is very similar to what we had in photosynthesis. You guys had NADPH in photosynthesis at the end of the thylakoid membrane, stealing those electrons to carry them off to the stroma. And here we have NADH, who's going to carry them to the mitochondrial membrane to jump through a very similar electron transport chain. pump some protons, drive chemiosmosis, phosphorylate ADP to ATP. All right. Notice at the same time we were adding in a little phosphate. Now this is the exact same thing on both sides. It just means that we've got the G3P and then the DHAP, which was converted to G3P, comes a little later, and it's going to follow the exact same pathway. So I'm just going to concentrate on this side over here. So we're going to steal some electrons. You need to know that out of the glycolysis, we are getting NADH. And notice that we are getting 2 NADH per glucose. Okay, 1 per pyruvate, but 2 NADH per glucose. That's going to matter. Okay, so we get some NADH. And once we've stolen those electrons and then added in this little phosphate group, we're going to have 1,3-bisphosphoglycerate, so 1,3-BPG. I really hope that sounds familiar to you because we saw that in photosynthesis. Then we're going to get 3-phosphoglycerate. Okay, notice the reason, again, it's called 3 is because of where these phosphates are. So why has it gone from 1,3 to 3? Well, because we took that phosphate that was on there and we have now given it to ADP and made our first ATP. Okay, so if you're looking at the differences between the molecules, that'll show you what's happening. Here's the phosphate has been ripped off. See, the phosphate is now missing here. Then we're going to go from a 3-PGA to a 2-PGA. All we've done is taken this phosphate and moved it up a carbon. So there it was here, now it's up here. So 3-PGA, 2-PGA, sounds like a countdown. We're going to lose a water here, and now we've got phosphoenol pyruvate, PEP. You also saw these guys in photosynthesis. PEP was the one that we saw when you were dealing with C4 and CAM plants, when they couldn't use Rubip to fix the carbon dioxide because of photorespiration, when the oxygen was getting in the way and stopping Rubisco from doing its job. Then we had to smush together this phosphoenol pyruvate with the PEP carboxylase, and we were stealing that, storing the carbon dioxide as malate and using it later when the conditions were just right. So we were flooding the system with carbon dioxide that way. So we're seeing PEP again in a reverse order here, okay? So we've stolen an ATP here, and then off the phosphoenol pyruvate, again, it's called phosphoenol because of how that phosphate is attached to the pyruvate. Once you steal the phosphate, you're just left with normal pyruvate. So this is phosphoenol, this is just normal pyruvate. Done, okay? So again, major things here: glucose, 6 carbons, activate with your phosphates, falls apart into DHAP and G3P. This guy gets converted to G3P, steals some electrons, makes some ATP, shift, makes some ATP. I end up with pyruvate. And again, I've done that twice. So if you look, you have created 4 ATPs. You end up with 2 pyruvates and 2 NADHs. You need to know those major things coming out of this thing. So because we've got our glucose and we put into ATP, but we got out 4 ATP, when we simplify this equation, you can see we actually only have a total net. We only gained 2 ATP from that process, okay? So that's the same thing with the ADP that we put in. We put in 4, but we got out a couple. The equation's going to reduce down to 2. So this is what you need to know. You have 2 pyruvate 3-carbon molecules at the end, 2 net ATP overall, and 2 NADHs. The NADHs are going to become very important because as electron carriers, they're going to make the massive most amount of actual ATP through oxidative phosphorylation. Let's go back up to the first page now. So here you've got your glucose, 6-carbon, broken halfway into 2 G3Ps, and then finally down into the pyruvates. That's just counting the carbons. And again, 2 ATP were used for activation, and 4 were produced, so we have a net of 2. And then we have 2 NADHs, and again, it's going to give its electron away. All right. This is an example of substrate level phosphorylation. Substrate level phosphorylation. So what do we mean when we say that? Well, substrate level, again, is when you have an enzyme that rips a phosphate off of an already existing molecule. And that molecule is called the substrate. It goes into the enzyme steals that phosphate and gives it to an ADP to make this molecule right here. And the difference between ADP and ATP is just how many phosphates they have, whether you are a triphosphate or only 2 diphosphate. It's the only difference. And again, this part of the molecule here is extremely similar to what you see in a nucleic acid. The adenosine, the base plus the sugar is the same. The adenine base is the same. The idea of having a phosphate attached is the same, but having two or three phosphates is what makes this an energy carrier molecule rather than an information storage molecule. So quite interesting that those molecules are very, very similar to do pretty much two of the most major jobs in the entire organism. Pretty cool. Then we've got the production of two molecules of reduced NADH, again, because it's stolen electrons. And it's going to move to the electron transport chain, which is inside the mitochondria, just mito for short. Electron transport chain, ETC. And we're going to give those electrons to the membrane. They're going to bounce through just like they did with photosynthesis. Different players, different cytochromes and proteins in the membrane, but the same idea. I'm going to pump protons to one side of the membrane. That's going to drive chemiosmosis with that proton gradient. And then I'm going to be able to run those protons back through an ATP synthase and crunch together ATP. And that'll be called oxidative phosphorylation. So the stuff that is made here, these 4 ATP that are made in this particular process, I'll scroll back down again. Notice that the phosphate was already on a molecule. It was already on this substrate and given to ADP. That's what makes this substrate level. Any ATP that is produced through chemiosmosis is oxidative phosphorylation. Okay. Then what you can do with the pyruvate once glycolysis is done is you can either run through the aerobic pathway, which means, in other words, the pyruvate's going to go into the mitochondria and get processed, or you can do the fermentation that we talked about last day. Depending on what type of organism you are, you can do lactic acid fermentation, ethanol fermentation. There's a bunch of others out there as well. I'm not going to go through every type of fermentation. These are just the 2 most common ones that'll help you for university in the fall. All right. So that's it. That's glycolysis in a nutshell. Make sure that you go through and you give yourself a good understanding of this molecule to that molecule to that molecule and so on. It'll help you out when you're actually getting into the nitty-gritty details when I start to do some assessment of what you guys have learned. But the main most important thing is that you know, let me scroll back up, that you know it's glucose to G3P to pyruvate. There's 2 of them. You're going to create 4 ATP, but you put 2 in, so it's a net of 2, all right? And that you are producing 2 NADHs. If you can memorize those suckers, that is going to be the most important thing to know by the end. It occurs in the cytoplasm. It's anaerobic, all right? Just want to point out before I finish that these 2 waters right here are not waters that we count in the overall equation for cellular respiration. These ones don't count. There's water in and out all over the place that we haven't mentioned because we're doing a lot of hydrolysis reactions. So when we're producing water or when we're using water, until we get to the very end when you see oxygen being used, know that all of these waters are going to cancel each other out in the overall equation. They're not going to be counted as the 6 that we talk about when we're doing that overall equation. All right. Well, good luck studying. If you have questions, email me and I'll try to get back to you as soon as I can. Have a good day.